Hi everyone, welcome to my channel once again. Today I am going to take you through a machine learning project that I worked on last week. So I thought I would share the result and the entire procedure with you, my viewers, especially those who are interested in machine learning and those who want to develop expertise in the field of machine learning and data science as a whole. Now with machine learning projects, you obviously need data. So for this project, I use RNA-seq data. RNA-seq is one of the sequencing technologies that is used in the field of biology. So you can look it up and just search RNA-seq. You have lots of articles and tutorials about this particular technology. So just look it up. So to put it simply, this data set I use is a kind of bioinformatics data. So this data comprises of gene count values and then cancer types. And so what I mean is that this data set has a measurement for gene count values done for different cancer types. We will look at the cancer types later on in the tutorial. The original data set can be found here. Just follow this link here. I'll also leave it in the description box. But I downsample the data, so I use just a subset of it, which can be found on my GitHub repo. So this is my repo, by the way. I will leave this link in the description box. So just go to the data folder here, and then you will see the data I used. It's cancer gene expression. So that's what I use. So the original data can be found here. Just follow this link and then you get to this page here. So here you can get the full data that I used. Okay, now let's proceed. So let me just close this tab. Let me just put it somewhere here. Yeah, so back to our notebook. So this particular project was done in Python. And these are the libraries that you require if you want to run this notebook on your laptop, on your PC. You can just do that. So just come to my repo here. Let's get back. And then you will see notebooks here. This folder here, just open it. You will find the notebook here. You can just download and then run it. So just do that. If you don't have Python on your PC or if you don't have the libraries, don't worry. You can run everything using your browser. Okay, so just use Google Colab. I will show you how that is done also. So if you want to run this notebook on your PC without having it done locally, you can just come to Google Colab. I will leave this link in the description box for you as well. So visit Google Colab here and make sure you've signed into your Google account. It's important. So once you are there, come to this tab here, GitHub. Click on GitHub and then paste the link here, the link for this repo here. Just paste it there and then just search. Just click on search. And then once you search, you have the notebooks appearing here. So just scroll through until you get to the right notebook. So the notebook we are going to use for this tutorial is just this one here. This particular notebook here called notebook/cancer prediction using gene expression data. So this is what you need. So just click it. And then once you click it, you have it opened for you. Okay, so there's another way you can use to run this particular notebook. So just try your hands on them and then we can proceed. So now we can proceed with the exercise. So ladies and gentlemen, welcome to the tutorial. Okay, so we are going to start the exercise. Let me switch to this notebook here. I just slide this text. Okay, so the next thing we are going to do is to import our libraries. So these are the codes for importing the libraries. You also notice I have some comments here telling you what each group of codes is doing here. And so these comments here are also activities that when you are performing a machine learning task, you will do. These are things that you have to do. So you have data handling, visualization, preprocessing, feature selection, classification, and then evaluating the model's performance. So these are things you need to do. So just take note of that. So let's import our libraries. So I'll just highlight this cell here. I'll just run it. Perfect. It's now time to read the data. So I have the URL for the file here. So let me also say that with pandas, you can actually specify a URL to a file. And pandas will get that link and then use that to read the file for you. So This is handy if your file is on a remote server, so you don't need to download the file, you can just specify the link. So here I am using the data here, and so I just specify the URL here, that's the download link, and that is what we have here. You should also know that with pandas you can read compressed files, so files like those with a .zip extension can be read without having to decompress the file. So you can do that with pandas. So that is why I have it here. Okay, perfect. So let's proceed. So I'll just run it to read the file here. I'll just use this to do it. Perfect. So it's also done. Now it's time to do exploration. So we explore the data so that we're able to get information. The information we get is useful. It will help us to understand the data, and it will also even help us to determine what methods to use for the downstream analysis. And of course, as we are exploring, we also need to clean the data. Cleaning will ensure that unwanted information is removed, and it also helps to properly format your data. So these codes here will help us do that. And I have some comments here that gives some description of what each code is doing. So there's more like a self-explanatory notebook that you can use. So just run them to get your outputs. So we will start by getting the shape of the data frame. Once we read the file here, we get a data frame. Data frame comprised of rows and columns. So let's run this, and then we have the result here. So we have 801 rows and then 8001 columns. Now for this data here, the rows represent samples and then the columns represent features. But of course, there is one column here which we don't consider as feature. That column is the label column. So that one is for the cancer types, the information about the cancer types. But the other columns are features that we will need. So take note of that. So let's get some detail here. Let's get the column names. We want to get some of the column names. We are looking at the first, second, and then the third columns. We use this code to get it. So run it and you get the column names. So I made mention earlier that we have gen counts for different cancer types. So the columns have also been named. So we have gen 1, gen 2 up to the gen 8000. That's the 8000th gen. So that's what you need to know. So let's look at the next activity. Let's get the name of the column. We want the last column. So let's run this to get it. So we have the column name is cancer type. So this is what I was talking about. So there's the label for the cancer cases. So the column here will give the cancer type for each sample. Perfect. Now with machine learning, one of the key things you have to do is to check for missing values. The issue of missing values can affect the model performance. So you need to check and then account for it or correct for it. So this code here will help us do that. So what this code is doing is trying to get all the columns and then check if any cell under that column has its value missing. Basically, that's what it's doing. So that's what we do. So we just check and then get the number of columns with missing value. That is what we are doing here. So run it, and then we have the output here. So we have the output here, and then the output here says the columns with missing values is 0. So that means this data is clean. We don't have any missing value, which is very, very important. So we need that information. Perfect. So let's proceed. So if you've done it up to at this point, I would say good job. That's what this text here is for. So just proceed. Now we want to get the counts. It's important we get the counts. When I say count, we want the count for the various cancer types. Let's look at that here. So here we will get each of the cancer types and look at how many samples were diagnosed with that particular cancer. So this code will help us do that. So run it and you will get the information here. So we have 5 different cancer types. So that's one. So these are the labels. I think this one is for breasts, and this is for kidney. We also have lung. We have, I think this prostrate, and I think this one I'm not really sure, then I'll just check. I'll give the full description for these codes here in the description box. So these are all cancer types. So these are cancer cases for different organs of the body. So just take note of that. With this count here, we can make a meaning out of it, or we can easily understand what is going on in terms of distribution by visualizing it. So we can do a bar plot here. So run it and you get the information here. Okay, so we have our bar chart here. Now with pandas, you can visualize your data in other forms as well. So let's say you want to display the class distribution here in the form of a pie chart. You can do that here as well. So you just have to modify the command and say kind equals pi. And once you run it, you have your pie chart displayed for you. You can even modify the figure size by specifying this here. Fixed size equals, and you specify the dimension that you want and then run it. So the figure size will help you to modify the size of the figure. So just take note of that. You can explore and then just look at what works best for you. Just check the online documentation and you have lots of options available for you. So let's do that. Let's reverse back to the bar chart and then proceed. Now if you look at this bar chart here, it's easy to compare. It's easy to compare the values for the different classes or the different cancer types here. Now you will see that there is an obvious difference. BRC, that is the breast cancer class here, there are high proportions of it in the data, and you can see that by looking at the height here. So just compare this and then this one here. So you see the difference. Now this observation here is what is referred to as a class imbalance, where there is a high proportion of one class than other classes in the same data. So it's important to know that. Now, the class imbalance situation can affect the model's performance. So you need to make sure that you have resolved it. And you can only find out if there's a class imbalance when you explore the data. And that is what we are doing now. Some methods account for class imbalance, and then for some too, they don't account for it. So we need to find ways to correct this particular issue. And so there are methods to deal with class imbalance. You can do oversampling, or you can also use undersampling techniques. But these are things we'll look at in future tutorials. But for now, the assumption is that the methods we are using are suitable for data sets which have this class imbalance situation. So let's proceed. So let's move on. The next thing we are going to do is to perform data preprocessing. Data preprocessing helps us to properly format the data. It helps us to put our data in an appropriate format that the models can use. And we will start by separating our features from the class or from the label, and we do that using these commands here. We are dealing with a supervised learning approach. So we just separate the values here, which are the features from the labels here. So let's run this to do that. Perfect. Of course, you can also do some exploration here. Let's say x.shape, just to see what we have here. We can do y.shape, just to see that also here. So things are taking shape nicely for us. Let's proceed. The next preprocessing step is to encode the labels. So yeah, let's go back to our cancer types here. You see that we have them here. These are categorical data. Now with machine learning models, they require that they require that we feed them with data in numeric forms. And so if the data is categorical, like in this case, then you need to convert them to numeric forms. And that is why we are doing the encoding. So we are encoding the labels. So we will give them integer values. And that is what these codes are doing for us here. So let's do the encoding. Let's run this command here to do that. Perfect. So we have it done. So just check. So the labels here, these are the original values. We have them here. So after encoding, we have them as integer values, and these are the ones here, classes. So classes, and you have them here. So this is what we are going to input or feed or pass to the machine learning model alongside the features. So we have 0, 1, 2, 3, 4. We have them here. So these are arranged, I think they are arranged in alphabetical order. So you can just map them using these values here. So 0 for BRCA, 1 for COAD, etc. So that's what we have here. So just take note of that. So let's proceed. So the next thing to do is to split the data. We are going to split into training and then test subsets. So why are we doing the splitting? We need to let the model identify patterns. We also need to test the model to see whether it performs to our expectation. And that is where the training and the test subsets come in. So the training subset will be used initially. So it will be fed to the model, and then the model will use that to learn and identify patterns. And these patterns that it identifies, or this experience it acquires, will then be used to make future predictions. It will be used to make predictions on data it has never seen before. So that is what we are doing now. We are splitting 80-20, meaning 80% of the data is for training, and then test sets will have 20%. That is what we are doing now. So we use this command to do the splitting. Okay, let me just put here. Okay, so we have that done. We will move on to normalization. Yes, this is also an important step. Before we even talk about normalization, let's just do a quick exploration here. Let's just do this. There's something I have to show you first. Let's say X. Yeah, let's use the original data. X data frame. I'll just use a subset of it to demonstrate something for you now. It's just a quick statistics about the data. Perfect. If you look at this here, you'll notice we have some statistics here. We have count, mean, standard deviation, minimum, up to maximum. We will focus on the minimum and then the maximum values. Take a look at Gen 1. We have 0 and then 1.4. That is the range, 0 and then this. We have minimum, maximum. Take a look at Gen 2, 0, 6.5, and then the same for the others. Okay, now you realize that they are in different ranges. That's what you need to know. They are in different ranges. Okay, we also have Gen 10 here, 1 and then 4. So when we have an issue like this and then we proceed with the data as it is, it will affect or may affect the model's performance. Okay, because we have different ranges. So usually what we do is to make sure we have the values in the same range or in similar ranges so that the model can use them and kind of put them on the same level or on the same scale, and then make easy comparisons. And that is what this normalization will do for us. Okay, so that's what we are going to do. So let's proceed. So these are the codes that will do the work for us. It's going to scale the data between 0 and then 1. Now, another reason why we do this normalization is that we also want to avoid bias. If you don't normalize, the models may return results which will project that some features perform better than others, or some features are more important than others, or they are more relevant than others. Okay, so this bias can be avoided if we scale the data and make the values have the same range, and that is what we are doing now. So let's proceed. So we will just scale between 0 and then 1 using min-max scaler. This is also one of the scikit-learn models. So let's run this. So run this, and then we have the data scaled for us. Let's just check and then just make sure everything is correct. I've also given them different names for the normalized values. So let's just do a comparison. Let's say the original X train here, let's just say string dots. Let me just check the type first. I'll just say string. I just want the first. Okay, I have 0, 0, and then 3. I'll just put it this way. So let me just confirm the data type first before I make the query. String. Okay, so this is a data frame. Okay, perfect. So I'll just say string.iloc 03. I just want this particular cell, the value for this cell. So the value for this cell is 2.18164, etc. Let's look at the normalized value. That one is a numpy array. So we have string norm for normalized. So 03. Notice the value here. So this is what? Between 0 and 1. And that is what I am talking about, the normalization. So the data has been scaled. So all the values are between 0 and 1. It's important you do that. Okay, so some will do normalization, some will also do standardization. So just look at what method works best for you. But for this tutorial, we are doing normalization. So take note of that. Now, let's proceed. The next thing we are going to do is to perform feature selection. Feature selection will help us to select the relevant features for classification. For this tutorial, we are going to use the mutual information algorithm here. So what this algorithm does is that it computes the scores for each of the features. So here, the scores refer to their respective importance or relevance. After that, we are going to sort the scores and select the top n features. Here, we are going to select the top 300 features. And so let's proceed to do that. So I have my mutual information model here. So I'll run it to get the scores. So let's run it. Okay. So after running, we are going to get the scores here. And then we will define the top 300 features. And then we are going to sort and select the features. So here, the numpy.act sorts the data and then also keeps records of the indexes of the features. And so once we have the records of the indexes, we can easily map the scores and also map the features themselves. So that is what we have here. And so we sort, and then after sorting, we reverse so that we get the highest to the lowest. And then we can select the top n features here. That is what this code does for us. But before we even do that, let's take a look at the scores here. So let's do it here. I'll first do the shape here. So you will see that we have 8,000. There is a numpy array. We have 8,000 because we are dealing with 8,000 features. So let's look at, let's say, the first 5 scores. Okay, so we have them here. So at the moment, it has not been sorted. The sorting will be done here, but these are the scores we have. Okay, so you should also know that with the scores here, these scores are also presented in the same order that the features were fed to it. So once you know the features, you know the order, you'll be able to map them to their respective scores here. So the features, let's just take a look at the features here. So let's say features equals string dot columns. So the features should also be 8,000. Let's do a shape and we have it there. So let's look at the first 5 features. We can say features and we have it this way. And so once you have it this way, you can easily map the features to their respective scores here using their indexes. That is what is being done here. Okay, so let's pick the top 300 features. Let's run this code here. Okay, so it has been done. So once we have the indexes for the top 300 features, we then use the indexes here to select them. And that is what we have here. So let's run this to get the subsets. So now do a shape of the variables here. Let's try with this one here. And we are going to get 300 because we are selecting the top 300 features here. The number of samples will remain the same. Let's try with the test as well. And that will also give you 300. So that is how we do the feature selection here. So now let's proceed with the rest of the activity. Okay, so we are now on the classification part of the tutorial. The end goal of classification is to let our model be able to tell us that this particular sample we gave it belongs to class A, class B, or class C. In our situation, we are dealing with cancer types, so the model should be able to tell us that this particular sample here belongs to this particular cancer group, or this group, or this group. That is what we want at the end of the day. There are several models available for performing classification tasks, and one of them is random forest classifier. And that is what we are going to use for this exercise. I am using random forest classifier because it works with multi-class and it also works with high-dimensional data. Now with the classification, it is important that before you use any classifier or any model, you read about the model, get the strengths and weaknesses, and that will help you to determine if the model is appropriate for your work or not. You need to get as much information as possible, and that will help you to get the appropriate models for your work. Some of these things, they come with experience and experimentation. So just experiment, read, and then just practice. And that is why this tutorial is being made for you. So let's proceed. With the classification, we are going to do two things here. We are going to train the model and we are going to test the model. So let's start with the training here. So with the training, what we do is that we feed the model with the training data. This is to help the model to learn and identify patterns, and the knowledge and experience the model gets will be used for making future predictions. The predictions will be made on the test data set. So let's proceed. So this is my random forest classifier here. This is a classifier, and this here is a parameter that we can change. Now this page here gives you information about the random forest classifier, so you can just visit this page. I will leave the link in the description box. So this is the scikit-learn page here. So all these parameters can be changed. We call that hyperparameter tuning. That is something I'll cover in another tutorial. So let's get back to work. So there's a random forest classifier, and you will also notice that I have one versus rest classifier here. So one versus rest classifier here, I'm using that because of one of the metrics I'll use for evaluating the model. So one of the metrics I'm using was designed for binary classification. That's the ROC curve. So if we are going to extend it to multi-class problems, we need to find a way to make that happen. And so the strategy is that we pit each class against the other classes, and that is why it's called one versus rest. So at any point in time, we have, let's say, class A, and then the other classes, let's say A, B, C, D, they all become one class. So we have that class A against all these other classes. That becomes a kind of binary situation. So we do that for each of the classes, and that is where the one versus rest name comes in. So it is a way to extend the ROC curves to multi-class problems, and that is why we use this here. Perfect. So let's proceed. So we create the classifier, we have our strategy, and then we feed the classifier with the training data sets here. This is a supervised learning, so we feed the training data together with the labels, and that is what we have here, and then the model will do the rest. So after that, we let the model do its prediction. So that's the second part here. After training, we feed the test data to the model here, but this time we don't add the labels. We let the model do its prediction here, and that is what we will use to do the evaluation. We will compare the values that was predicted, or the classes that was predicted by the model, to the actual labels, and then we get the performance from there. We also have this line here, PRAD-prob. So this one here, we are given probabilities. We are trying to get probabilities for the classifier. So what we are doing, that these probabilities will be used to generate the ROC curves for us. That is what we are doing here. ROC or raw curves, I mean, any of them should be fine. So I'll just use them interchangeably. So that is what we have here. So we are going to run this to get a classifier started, and then we can now proceed. So the model has been trained, and that is what we need now. So now it's time for the model evaluation. So let's proceed. Okay, it is now time to evaluate the model. So here what we do is that we compare the model's predictions to the actual labels or outcomes. We already have the model's predictions here, and we have probabilities we will use later on. Now, there are lots of metrics available to evaluate a model. For this tutorial, we are going to use these metrics here: accuracy, precision, recall, F1 score, confusion matrix, and then ROC curve. Let's start with the first four. The codes here will generate the scores for us. So let's run the cell. Okay, so we have the scores. So we have them here. The 4 scores here. The scores range from 0 to 1. If you take a look at these scores, you will see that they are all approaching the value 1. And so here what I can say is that we have high scores. High scores indicates that the model is performing very well. And so in our case, the model is able to predict correctly most of the classes in the test data sets. That is what it means. We have high scores, all right, but it is important to also do further checks to ensure that the model appears as it seems. But these are things we will do in later tutorials. It is also important that you check the model's performance on the individual classes, and so we use the classification reports to do that. The classification report will generate a table for you, which is here. And then the table here has the classes and then scores. Okay, so we have precision, recall, F1 score. Okay, so you can look at these scores and also look at the classes, and you'll be able to know where the model is performing very well and then where the model is not. And so here, if you take a look at precision, for example, you can see that the model here has a high precision score for these 4 classes here. And then comparing these 4 classes to BRCA, we see that the model has a lower precision score for BRCA. And so in terms of performance, the model does better in predicting these 4 classes than the BRCA class. That is what this means. You can also check for recall and then F1 score. The support here tells us the number of actual instances for a particular class in the data, but our focus is the scores here, so we will not go into details with that. Let's also look at macro average. So the metrics we are using were designed for binary classes, and one way to extend these metrics for multi-class problems is to compute the macro average. That is what this here tells us. Okay, so we have that there. Weighted average here accounts for data imbalance. If you check the codes, some of the codes here, I have average equals weighted. Okay, so from the scikit-learn documentation, average equals weighted is appropriate for class imbalances. And since we had a class imbalance, it was important that I added this particular option here. Okay, So let's proceed. So let's go to confusion matrix. So confusion matrix here generates a table that has correct classifications and then misclassifications. So let's generate the matrix and then you'll see what I'm talking about. So let's run this code here. This will generate the matrix and then I'll put them in a data frame here. And then I'll display it here for you. Okay, so I have the table here. So this side here is for the actual labels or true labels. Here is predicted labels. So let's look at BRCA. So the number here tells us how many BRCA cases were also predicted by the model to be belonging to BRCA. So that is what we have here. So the 60 here means that there were 60 cases of BRCA and the model predicted all these 60 cases to be belonging to the BRCA class. So in other words, we have correct predictions here. None of the cases was predicted as belonging to other classes. So here we have 0. And so here it means that the model was able to predict correctly all the BRCA classes. When we come to COAD, one COAD case was predicted as BRCA. And so here, this is a misclassification. When we come here, 15 COAD cases were predicted correctly. Okay, that is why it's also under this column here. Now, there are a number of reasons why we can have misclassifications. One of them is outliers. When there are outliers, it may affect the model's performance. And so it will lead to, it may lead to misclassifications, and that is what we are seeing here. So it's possible that this particular case here was an outlier. Misclassifications can also occur due to errors in terms of labeling, the human elements. It could be possible that maybe during the data collection, the labels that was given were actually wrong, and the model has rather made a correct prediction. And so the predictions made by the model and the ones we have in the data will be different. So that's also another cause or another possible cause for misclassifications. So we take note of that. There are other causes or possibilities, but these are things we will look at in later tutorials. KIRC, all predictions were correct. The same for LUAD and then PRAD. Okay, so we have a good model here. And actually, it's not surprising because we have high scores here. Okay, let's proceed. So with the confusion matrix, you can also visualize it in the form of a heat map. So here I use Seaborn to generate a heat map here. That is it. So the heat map, you have a color code to give you the numbers, and then you can use that to infer, okay, where the model is doing well and where the model is not. Okay, and you can also infer the number of correct classifications and the number of misclassifications. So we take note of that. Now you can also generate this heat map without having to generate the confusion matrix table here. So scikit-learn can help you do that. So the plot confusion matrix can do that for you. So let's run this also. Okay, so we have that also there. So we have the same observation here because it's obviously the same data. So take note of that. Now let's go to the last evaluation method here. That is the ROC curve. So ROC curves are used to visualize the performance of models. And so here I will generate a curve here. Now what we are doing is that because we are using the one versus rest approach, we need to also format our predicted values. Okay, we need to format it so that it has the same shape as the probabilities. If you take a look at the probabilities, let me just show that here. Probabilities are generated for each class. So let me just do a shape here for you to see. We have 5. So this 5 here probabilities are generated for each of the classes, and that is why we have them in this particular shape. Okay, but with our current class, the predicted class, let's just go back to check the name. We have YPRAD. Okay, yes, so YPRAD here, if you check the shape, it's just one column. And this, when we use this, we may have some errors because we need to have corresponding probabilities. Okay, each class should have its corresponding probabilities, and that is why we bannerize it here so that we can get it in that particular format. And so take notes of that. And so we do this to get the formatting done, and we can now feed this particular value here together with the probabilities, and we can get our ROC curve. So the ROC curve will be generated, and then AUC values are also generated using these codes here. So let's run the cell. Okay, so we have the ROC curve generated. Now the ROC curve you see here, yes, so you may not have seen this kind of curve before. Usually you will see it this way, but this is also a curve. And so what we have here is that we have high AUC values. Because the model is performing very well, the AUC values are all 1, I think, with the exception of this one here. And so if it's 1, it's a perfect score. So you have something like a straight line here. And so that is why all the classes here, you have them here. You have the curves all here. Okay, so if it were to be, let's say, 0.8 or maybe 0.82 or thereabouts, you will have them this way. But because the model is performing very, very well, that is why they are all here. So this is also something that you can find in machine learning modeling. So you take note of that. And so just take note of that. So once you are done, you can save your plots. I'll just right click and then just save as. I'll save it here. Let me see. See, this confusion matrix. I'll save it. You can also save the others as well, and then you can put them in your reports. Okay, you can also save this as a data frame and then open it with a spreadsheet software. So those are things you can also do. So that's it. So this is how we end the machine learning tutorial for today, and I believe it has been helpful. And so if you have any comments, if you have any points that you think is useful, you can just put it in the comment section. We can all discuss it together. If you also want to support this channel, you can buy me a coffee. The link is also there. You can just do that as well. And then we proceed. So I also have a presentation on this same tutorial. So here it's more like an overview of everything, and I give some additional explanations here. So you can check that video out. So that'll be all for this tutorial, and then I'll see you in the next session. Goodbye.